Hello everyone, welcome to today's lecture. I am again Qazi Fariduddin, representing Department of Botany. I extend a warm welcome to all of you for this plant physiology and metabolism course. In today's lecture, we will dive in fascinating world of light reaction of photosynthesis. Now, what is the basic mechanism of photosynthesis? You see in this figure, it is evident that photosynthesis is an oxidation reduction process where oxidation of water, that is electrons being removed from the water, is generally coupled with the release of oxygen and reduction of carbon dioxide, which leads to synthesis of carbohydrates, as we have discussed in detail in yesterday's lecture. It is generally a two-stage process. During stage 1, known as light reaction, generally photolysis of water takes place. In subsequent stage 2, electrons removed from the water are then used to reduce carbon dioxide, known as CO2 assimilation. So basically stage 1 and stage 1 is photochemical, however stage 2 is purely chemical reaction. Also the electrons in these photosystems lead to synthesis of ATP and NADPH, which are then further utilized in the assimilation of carbon dioxide in the Kelvin cycle. Now we will discuss light dependent photochemical reaction, that is the first step of photosynthesis, which are basically dependent on the availability of lights. So what exactly are these light reactions? Question arises, what exactly are these light reactions? Why are they termed so? So the light reaction can be defined as the process of photosynthesis that converts energy from the sun into chemical energy in the form of NADPH and ATP. Basically, these light dependent reactions use light energy to make two molecules required for the next stage of photosynthesis. One is the energy storage molecule, that is ATP, and other is reduced electron carrier, namely NADPH. Generally in plants, these light reactions takes place in thylakoid membranes of organelles called chloroplast. You might be aware. Also, the process of formation of energy source ATP from ADP and inorganic phosphate during light reaction by utilizing light energy is called photophosphorylation. Basically, there are two types of photosystem, photosystem 1 and photosystem 2. Both photosystems contain many pigments that help to collect light energy, as well as a special pair of chlorophyll molecules found at the core, that is at the reaction center of the photosystems. The special pair of photosystem 1 is called P700, while the special pair of photosystem 2 is called P680. Now, what are the important events that takes place in light reaction? First, absorption of light energy by two photosystems, then non-cyclic and cyclic electron flow. Splitting of water in non-cyclic electron flow generally takes place, and ATP is synthesized via chemiosmosis, and finally NADPH formation takes place. Now we will discuss each of these aspects in detail. If we divide this primary photochemical reaction or light reaction into different heads, in the first step, the energy will be absorbed by these photosynthetic pigments, which we have discussed already, that is photosynthetic pigments such as chlorophyll A, chlorophyll B and carotenoids. These are found in the thylakoid membranes of chloroplast. and these are light harvesting molecules. Now in the second step, transfer of this energy from accessory pigments to chlorophyll A. When pigments generally absorb a photon, it is raised to an excited state as evident in the figure, meaning that one of its electron is boosted to a higher energy orbital. Now these pigments in a photosystem acts as an energy funnel, passing the energy towards the main reaction center. When one of these pigments is excited by light, it transfers energy to a neighboring pigment through direct electromagnetic interaction in a process called resonance energy transfer. The neighbor pigment in turn can transfer energy to one of its own neighbors. In this manner, this process is repeated multiple times. In these transfers, what happens? The receiving molecules does not require more energy for excitation than the donor, but may require less energy, that is, absorbs light of a longer wavelength. Therefore, pigment molecules collect energy that transfer it towards a central part of the photosystem called the reaction center, that is chlorophyll A. Now, both the photosystems contain these pigments that help to collect light energy as well as a special pair of chlorophyll molecules found at the core of the photosystem, that is reaction center. The special pair of photosystem 1 is called, as I mentioned earlier, P700, while the special pair of photosystem 2 is called P680. However, chlorophyll can also absorb light energy directly. Now the third stage, activation of chlorophyll A by photons of light. The reaction center of both the photosystems contain a unique pair of chlorophyll A molecules, which are considered a special pair. When the chlorophyll molecules of both the photosystems receive a photon, it stops being transferred to other pigments through resonance energy transfer. Instead, it gets excited to higher energy state. So, the metastable triplet state is the main excited form of chlorophyll that actually takes part in further photochemical reactions. Now it releases energy along with an electron, therefore attains a sort of positive charge and become oxidized. Now try to understand, this electron is then passed to another molecule in the complex known as primary electron acceptor, and once this transfer happens, the electron starts its journey through an electron transport chain. Now very important event in this is Hill reaction in this sequence. Now question is, what is Hill reaction? So Hill reaction basically involves the light driven transfer of electrons from water to Hill reagents, which are non-physiological oxidants. And this process basically was discovered by Robert Hill in 1937, which revealed that the oxygen produced during photosynthesis through the oxidation of water occurs independently of the mechanism that converts carbon dioxide into sugars. So basically Hill observed that isolated chloroplasts from the plants release oxygen when exposed to light in the presence of an appropriate electron acceptor like ferricyanide and ferric oxalate. Most importantly, the isolated material by Hill basically comprised of thylakoid membranes lacking the necessary enzymes for carbon dioxide fixation. So one of the physiological oxidants utilized by Hill, now known as Hill reagent, that is evident in this slide, namely a di-2,6-dichlorophenol endophenol, which appears blue in its oxidized form and colorless in its reduced form. Subsequently, S. Oka demonstrated that NAD serves as a biological electron acceptor in the chloroplast. NADP, which in turn acts as a reducing agent for carbon fixation in Kelvin cycle. Now, moving onward, we will discuss how non-cyclic photophosphorylation takes place. Once light energy is absorbed by the pigments and then by photosystem P680, the high energy electron transfer is now passed to an acceptor molecule. So, this is positively charged, which is P680, and a strong P680 serves as a strong oxidizing agent that needs to be re-reduced and thus attracts an electron from an electron donor so that it regenerates the original P680. So, water basically acts as an electron donor in the oxygen photosynthesis, you might be aware. Two water molecules are split to yield generally 4 electrons and 1 molecular oxygen, as you can see in the reaction. Therefore, this splitting of water releases the oxygen that we breathe, human beings generally. And this photolysis of water is catalyzed by a protein complex called the oxygen evolving complex, which is generally present on the luminal surface of the thylakoid membrane. The oxygen evolving complex contains several proteins like one manganese stabilizing protein, PSPBP and PSBQ, as well as 4 manganese, calcium ion, collide ion and bicarbonate ion. So, the electrons released from the splitting of water, 4 electrons basically derive from the photolysis of 2 water molecules, do not directly transfer to P680, that is in excited state, as this complex can only accept 1 electron at a time. You must note this at this juncture. Instead, the oxygen evolving complex oxidizes water and subsequently passes 4 electrons one by one to activated P680 center. This complex basically undergoes multiple oxidation states while abstracting photons and electrons from water molecules, ultimately releasing oxygen into inner thylakoid space. Basically, the precise roles of calcium, chloride and bicarbonate ions in this process remain still not clear. But you see that the important point to note here is that although each photon absorbed by P680 transfers only 1 electron, 1 electron and 4 photons are required to facilitate the generation of single oxygen molecule from water. This mechanism raises the pivotal question or very pertinent question, how these 4 photons synchronize to produce oxygen molecule. So, there exists a sort of mechanistic analogy involving gear wheel like structure that gathers the 4 electrons from 2 water molecules and sequentially delivers them to the reaction center. Furthermore, magnesium ions within the oxygen evolving complex cycle through 5 distinct states, as on the left side it is very clear different oxygen evolving states that goes from S0 to S4, that is it 5 states, each transition between these states constituting a photon driven redox reaction. The transition from S4 to S0 specifically results in the liberation of oxygen molecule. This is very important to note it here. And the comprehensive model for the photooxidation of water is referred to as S-state mechanism. The process involves 5 reaction steps that sequentially release a total of 4 water-driven photons into the thylakoid lumen. As you know that oxygen evolving complex doesn't directly transfer electrons to P680. So instead, an electron carrier known as YZ, which is basically derived from a radical formed from tyrosine residue in the D1 protein of the photosystem 2 reaction center, functions between the oxygen evolving complex and P680. As you see in the figure, the sequential absorption of 4 photons, each causing the loss of 1 electron from manganese center, produces an oxidizing agent that can take 4 electrons from 2 molecules of water producing oxygen, and the electron lost from the manganese center pass one at a time to a tyrosine residue, that is YZ, in the D1 protein. What is the mechanism of electron transport in non-cyclic photophosphorylation? Before going into details of Z scheme, we should know the difference and exact role of photosystem 2 and photosystem 1. Basically, photosystem 2 comes first in the path of electron flow, but it is named as second because it was discovered after photosystem 1. That's why it is known as PS2. So, the special pairs in the chlorophyll A found in 2 different photosystems basically absorbs distinct wavelength of light. Also, the photosystem 2 absorbs light most efficiently at 680 nanometer, and PS1 photosystem 1 absorbs light most efficiently at 700 nanometer. So, after the special pair of P680 in the photosystem second absorbs energy and gets excited to a high energy state, this excited P680 can now donate its electron to the primary electron acceptor pheophytin, initiating a series of redox reaction in the first part of the photosynthetic electron transport chain. And it is evident the primary electron acceptor of photosystem 2 is a molecule called pheophytin, which is an organic compound resembling chlorophyll. However, in pheophytin, 2 hydrogen atoms are present instead of magnesium atom. This modification basically enables pheophytin to have distinct absorption spectrum compared to chlorophyll, so that it could be an efficient electron acceptor. After releasing its electron, P680 becomes positively charged and requires a new electron. This electron is obtained through the splitting of water molecules by a part of photosystem 2, as we have discussed in detail. The manganese center binds to 2 water molecules simultaneously, extracting 4 electrons, releasing 4 protons and producing a molecule of oxygen, as you see in the figure. Very interesting fact that around 10% of the oxygen produced is utilized by mitochondria to support oxidative phosphorylation, while the remaining oxygen is released into the atmosphere and utilized by the aerobic organisms for respiration. After pheophytin, electron transferred to plastoquinone. Plastoquinone exists in photosystem 2 in 2 forms basically, QA, which is tightly bound to the D2 protein, and QB, that is loosely bound to D1 protein. Plastoquinone serves as the active quinone in photosystem 2, while QA is produced to form the plastosemiquinone. QB can accept 2 electrons and 2 protons, resulting in fully reduced plastoquinol. Plastoquinol now then diffuses within the thylakoid membrane and binds to cytochrome B6F complex, which lies between photosystem 2 and photosystem 1. The cytochrome B6 complex oxidizes plastoquinol and pumps proton from the stroma into the thylakoid lumen. These cytochromes contain a heme group as a prosthetic group. In photosynthetic organisms, generally B and C type of cytochromes are reported. So this complex contains one Res-K iron sulfur protein, two cytochrome B molecules and cytochrome C1. The cytochrome B6F complex mediates the Q cycle, where electrons pass from plastoquinol to cytochrome B one at a time, leading to pumping of protons across the membrane. In chloroplast, the direction of proton movement is from stromal compartment to the thylakoid lumen, that is opposite to the mitochondrial proton movement. Cytochrome B6F then transfers an electron to plastocyanin, which is a small water-soluble copper-containing protein in the thylakoid membrane that shuttles electron between cytochrome B6F complex and P700 of photosystem 1. So, what happens during this electron transport chain? The electron moves from a higher to a lower energy level, thus it releases energy in the process. Once an electron has completed the first part of the electron transport chain and reaches photosystem 1, it combines with a chlorophyll A special pair called P700. However, because electrons lose energy along the way, they need to regain energy by absorbing another photon and become an excited state P700. Now, if we see in terms of source of electron, photosystem 2 obtains electron from the water molecules, which are split during the light dependent reaction of photosynthesis. On the other hand, the electrons that replenish photosystem 1 comes from an electron transport chain that originates from the photosystem 2. So, coming back, P700 after absorbing a photon becomes excited to a higher energy state, which is denoted as P700 plus, that is excited state. So, what happens now? Excited P700 now, which serves as an efficient electron donor, or you can say strong reducing agent. Now, excited electron is then transferred to primary electron acceptor of PS1, denoted as A0, which is modified chlorophyll molecule. This process results in the oxidation of P700 to excited P700, allowing it to act as an electron acceptor by accepting an electron from the plastocyanin. Now, the reduced form of A0, referred to as A1 phyloquinone or vitamin K1, then transfers the electron to a series of iron sulfur centers called Fx, Fa and Fb. After these, the electrons are then transferred to ferredoxin, which is a small water soluble iron sulfur protein present on the stromal side of the thylakoid membrane. Now, the soluble flavoprotein called ferredoxin NADP reductase catalyzes the transfer of electrons from the reduced ferredoxin to NADP, resulting in the reduction of NAD plus to NADPH. It's important to note here that 2 electrons are required for the reduction of each NADP molecule to NADH. This NADPH is essential because it provides electron to the Kelvin cycle necessary for converting carbon dioxide into sugar. So, in summary, during non-cyclic electron flow, PS1 and PS2 and PS1 are connected in a series. Excited P680 transfers electron to pheophytin. P680 is oxidized by light and re-reduced by electrons from oxidation of water. Pheophytin then transfers the electron to the plastoquinones Qa and Qb. The cytochrome B6F complex transfers electron to plastocyanin, which in turn reduces P700, and the acceptor electron from P700 is basically A0. The next acceptor A1 is a quinone. A series of membrane-bound iron sulfur proteins transfer electron to soluble ferredoxin. The soluble flavoproteins NADP reductase reduces NADP to NADPH. Now, for each electron transferred from water to NADP, 2 photons are absorbed, one by photosystem 2, another by photosystem 1. And to form one molecule of oxygen, it requires transfer of how many electrons? 4 electrons, 2 from water molecules to NADP. Hence, a total of 8 photons must be absorbed, 4 by PS2 and 4 by PS1. I will continue this lecture I will continue the same light reaction in my next lecture.